Hello everyone, my name is Tim Mitchison. I'm a scientist at Harvard Medical School, although at the moment I'm at, this is summer 2011, I'm at the Marine Biological Laboratory in Woods Hole, where I'm a Whitman investigator. And Woods Hole's a beautiful marine lab by the sea with a long history, and the topic I'm going to talk about today, self-organization in biology, it's particularly appropriate at Woods Hole since a lot of classic experiments were done here by Shinya Inoue, and I'll show you some movies that were taken here a while ago, and then some recent ones my group has taken here, working here as a Whitman investigator. And I'm going to talk today about self-organization in biology. Let me go to this sort of I'm going to start with a kind of introduction about self-organization as a kind of organizing principle in biology, as a way the genome gets turned into cells. And then I'm going to talk in slightly more length about a particular case study in self-organization, which is meiotic spindles. And in the first part, I'll be already, this is a meiotic spindle here, I'll be using this image quite a lot to illustrate my point. I'm not going to introduce the biology of meiosis and mitosis in the first talk. I just would like you to get a kind of feeling for what a self-organizing system is like. And so if I use language names of pieces of the spindle, I will introduce those properly in the second talk, where I talk more in detail about cell division. So here I want you to get a kind of feeling for the biology. And what I'm hoping to transmit is a sense of kind of wonder at living processes that I felt as a child, and I continue to feel watching living cells under the microscope go about the processes of life, and studying sort of how those processes work has been, my life's work and that of many other people. Okay, here's some outlines and key concepts. This is more for people who may be taking this as part of a course, but this is the things we're going through. I want to, let me just step back here for a second, and here's a diagram that I hope is familiar to any student of biology. Genes going to polypeptides, folding into proteins. This is the central dogma. This is the way the genome is read out, and I think nowadays we would add just another layer to this, so proteins can bind to each other and to other macromolecules like nucleic acids and assemble into very sophisticated protein machines. And at least to a first approximation, this genes into protein machines is something I think we understand conceptually. And perhaps the most sophisticated example of this, I'll call this self-assembly process, where genes rather directly encode the structure and dynamics of biological processes. This is a lovely example, bacteriophage assembly. So this is a bacterial virus down here. This is the genome of that virus. This is bacteriophage T4. This is taken from a classic review by Bill Wood. And you can see here in the genome where these different pieces of the bacteria are encoded. You can see over here they fold up into these polypeptides and stick together like a kind of very sophisticated Lego set, essentially. And you get this really remarkable machine here, a bacteriophage, one of the more sophisticated biological machines you can find. So this is a fantastic example of genes building a complex object. But I think many people looking at this bacteriophage would not say that it was really alive. It doesn't move, it doesn't change shape, it doesn't grow. The genes specify a very precise structure that does its job but can't adapt to changing circumstances and that kind of thing. And if I go forward one, I want to contrast that here to, I put in an amoeba here, growing and moving, and I think even quite a young child looking at one of these amoeba down a microscope, which is something I did with my kids' classes when they were younger. You can see an amoeba move even with your eyes, and everyone can tell it's kind of alive in some way. And how these proteins and proteins machines become living processes is really what I want to talk about today, mainly using the example of mitotic spindles, but trying to generalize a little bit from that. And in thinking about this problem, I want to highlight, I'll step back for a second here, a kind of scaling problem. How do proteins here, proteins and protein machines, which have nanometer diameters, and so they're quite small, how can they get together to organize cells which can be much larger? And the example I've chosen here is a particularly large cell, a frog egg, which is, I'll talk more about frog eggs dividing in my second talk because it's an experimental system we use. But you can see here the task of building a millimeter-style object, although microns would be more typical for cells, out of nanometer objects, and organizing them in space and time is kind of distinctly non-obvious how you would do that. And the ordinary principles of self-assembly that make protein machines like bacteriophages are going to be of limited help, I think. And sort of stepping back from this, I think a question I like to ask is how in general could one organize a cell, for example, so different bits of the cell knew what the other bits were doing and acted as a coordinated whole, as in that frog egg dividing, over many, many microns, a scale much larger than molecules. And I think there's, in theory, a variety of ways that this sort of could occur. One way that I'm going to really emphasize in my talk is making long protein polymers, joining proteins together end to end to make long filaments that can physically organize a cell. Another way I'll come to in my next talk, at least a little bit, is using chemical gradients. So a molecule that diffuses, is created in one place and diffuses across the cell is another way of doing it. A third way is using mechanical forces. If you were pulling more at sea, for example, A might detect that pulling. These are not mutually exclusive models. In fact, the spindle example I'm going to talk to you in some sense uses all three. And also, you might want to think about, are these the only ways you could physically organize the cells? I think there are probably some other principles. Think, for example, how a neuron moves information, which I won't talk about. I'm going to focus on this way of making large objects, protein polymerization. Here's the example of tubulin, which is the subunit of microtubules. And microtubules form by an end-to-end polymerization reaction. They break down again by a depolymerization reaction. This is a non-covalent reaction. This is the tubulin molecules stick together by the weak bonds that characterize all or most interaction between proteins, and they can form and dissociate quite quickly. So this in some way is a simple way of getting a much larger molecule out of a small one. And we can see here's an example in a rather small cell, a Drosophila tissue culture cell. You can see a radial array of microtubules kind of spanning the cytoplasm and organizing it. A more spectacular example, I think, is in a much larger cell, not a frog egg in this case, but a fish egg. I'll show you a movie taken by a student in my lab, Martin Woer, of microtubules in a fish egg dividing, a zebrafish embryo that's been transfected with a protein that binds to microtubules and then followed with a laser confocal microscope. And the field here is a whole 0.5 millimeter, 500 microns in diameter. So I can step back for this so you can see it. This is mitosis here is just finishing, and two radial arrays of microtubules are growing out and stretching through the cytoplasm. And these two sets of microtubules will specify where the cleavage furrow comes in, which is that dark line coming down. So the cleavage furrow is cutting down to the top, and it will know where to go because these microtubules will tell it. But you get the sense these microtubules grow through the cytoplasm and organize it. And we're actually spending quite a lot of time trying to understand these large asters in my lab at the moment. So I talked at the beginning in rather general terms about self-assembly. What I'm going to argue now is that microtubule assemblies, and particularly mitotic spindles, provide a great example of this kind of organization of molecules into living systems. And Shinya Inoue, who worked for worked still here at the MBL in Woods Hole, was one of the, I think, one of the great students of self-organization in the cytoplasm. And you can see here in this movie of jellyfish embryos dividing, mitotic spindles popping up in the cytoplasm and then going away again. The spindles, there's an example here. This is polarization microscopy. It's sensitive to aligned protein filaments. But when the movie rewinds, we'll just look at it just for a second again, but you'll see these mitotic spindles kind of magically appear, do their job, and then disappear again. And we'll talk more about that job of segregating chromosomes in a while. So there's the first mitotic spindle appearing and disappearing. Then we'll see the second mitotic spindle sort of magically to me appearing right, where is it going to come? Just right here, those sort of black and white quadrants there by polarization microscopy, and then disappearing again. And in a way actually inferred from this kind of microscopy that they're made of aligned protein filaments, which are in fact microtubules. And so that kind of spontaneous appearance and disappearance is one of the things we want to explain. Now, the simplest way of building a microtubule would be to have the tubulin subunits bind and dissociate as a forward and reverse of the exact same reaction with no other chemical change. This would be a very simple example of self-assembly, in this case creating an endless chain of molecules, but it's just like the bacteriophage, it's kind of the Lego block principle. And this simple sticking without any other chemical change, without any ATP hydrolysis, is the way most protein machines are built. And so this is sort of how you might imagine a microtubule being built. However, there's a problem with, that would occur for microtubules if the proteins polymerizing and depolymerizing were simply forward and reverse reactions of the exact same chemical reaction. And that is, once a microtubule assembled to equilibrium, if you looked at its length over time, it would remain very constant. There might be small fluctuations as subunits came and went. But in general, the polymer would stay at roughly the same length while the amount of tubulin in the solution was constant. And that would be a fine way to build a stable rod, for example, those bacteriophage tail fibers. But it wouldn't be suitable for building one of these mitotic spindles, which, as I showed you in the Inouye movie, has to form in a minute or two and then later dissolve again in a minute or two. So it couldn't work like this, just thermodynamically. And in fact, tubulin has evolved a way of getting around the constraints of equilibrium thermodynamics by coupling polymerization, this is polymerization here, to hydrolysis of a high-energy nucleotide, GTP. So tubulin polymerizes here in the GTP form. This is a growing microtubule. When the microtubule starts shrinking again, it's shrinking in the GDP form because you have a hydrolysis event here. And that means for every tubulin molecule that goes one full cycle here, you've hydrolyzed a molecule of GTP. That's 12 kcals per mole of chemical energy, something like that. And this kind of frees the microtubule from the constraints of thermodynamic equilibrium and allows it to do some more exotic things. And how does tubulin use that energy? One of the main ways tubulin uses that energy is to create a very large length fluctuations in the microtubule. So they grow and then shrink and grow and then shrink. And these are much larger and more rapid length fluctuations that would be possible just at equilibrium. And this process is called dynamic instability. And it's easier to actually appreciate this if you see a movie here. Here's a centrosome in frog egg extract, which I'll talk about in the next talk. You can see, I hope, individually lines growing out and then shrinking back. And each microtubule in this movie lasts of order 30 seconds or so. So the microtubules are growing out, sorry, I have to do it this way, growing out, shrinking back, growing out, shrinking back. And to do that, they need to burn GTP to escape the constraints of equilibrium. And although I won't talk about this, in fact, this evolutionary strategy for a protein polymer to couple nucleotide hydrolysis to interesting and complicated polymerization dynamics to allow self-organization and work is conserved amongst a number of cytoskeleton polymers in the tubulin and also the actin gene family. The tubulin family, which has both animal and plant eukaryotic homologs and also bacterial cousins essentially with the same fold, they bind and hydrolyze GTP during polymerization. In the actin filaments, actin in eukaryotes and PAR-M and other molecules in bacteria. They bind and hydrolyze ATP. But in both cases, the point mechanistically and biologically is that by coupling polymerization to nucleotide hydrolysis, you can get more interesting dynamics, more lifelike dynamics than would be possible at equilibrium. And it'd be fun to talk about how some of these other polymers use nucleotide, but I'm going to focus only on microtubules. But I definitely encourage people to read about the other ones. Okay, so we have these large length fluctuations, but how is that going to help us? How is it going to help the cell rapidly assemble a mitotic spindle out of soluble precursors, this kind of rather magical self-organization process? And I think it's fair to say we don't really know. That's still an open research question. I'll share with you one idea. It's an old idea. This is an idea Mark Kirshner and I developed when I was a PhD student in his lab a number of years ago. And that's that these length fluctuations, these large changes in length that are quite rapid, promote a kind of spatial exploration. They allow the microtubules to explore the space of the cell much more rapidly than would be possible without GTP hydrolysis. And I'll illustrate that with the example of hooking up a centrosome to the kinetochores of a chromosome. So microtubules are going to grow from the centrosome. They have to find these kinetochores. I'll explain why that is more in the next segment. But here, let's just say this is a challenging spatial problem. How can the microtubules do this? And the idea here is that microtubules are growing out, shrinking back, growing out, shrinking back, and exploring space randomly. Some fraction of them will hit the target here and become captured. And in that way, you can affect this connection without needing any other kind of information to be exchanged between the target and the centrosome. That's still, I would say, whatever this is a long time later, still an unproven idea. But I think the general idea of spatial exploration is likely to be correct. So what I want to talk about now, I'm not going to talk more about details of microtubules and how they work in this talk. I'll go into that a bit more when we talk in the next segment, where I talk more about mitotic spindles. What I more want to talk about now are what are the kind of general characteristics, the kind of lifelike properties, I'm going to call it, of a self-organized system. I already talked about spontaneous assembly from soluble precursors, this kind of magical just growing in the cytoplasm. I want to talk about two other characteristics, a characteristic but adaptable size and shape. I'll explain what I mean by adaptable here. And then also rapid recovery from damage. And I would argue, although I'll show only microtubule assemblies, that these are rather general properties of self-organized systems in cells. And let me start with adaptable size and shape. So that bacteriophage I showed you, which is, we can trace the assembly pathway directly from the genes, that has a very fixed size and shape that are directly encoded in the genes and the self-assembly pathway. For microtubule assemblies, although they have a certain shape, the characteristic shape, let's say, of the mitotic spindle, the size is in fact can adapt to the size of the cell, unlike the bacteriophage. And I'll show you an example here with cleavage divisions in a zebrafish embryo. This is actually a continuation of that movie I showed earlier at the one-cell stage. I'll step back here so you can see it. That was the second mitotic spindle. Now you'll see the third mitotic spindles, and as they go on. And as you can see here, as cleavage makes the cell smaller, of course the mitotic spindle has to get smaller, otherwise it would be too large for the cell. But I hope you can appreciate the way here that these self-organizing structures, the mitotic spindles, are able to adapt naturally and rapidly to a change in the physical circumstance, in this case size. Let me show you another. It's a kind of related in my mind is recovering from damage. And this is a meiotic spindle here in a parchment worm egg polarization microscopy. This is a lovely example from the work of Ted Salmon when he was a PhD student with Shinya Inoue. This movie, I'm pretty sure, would have been taken here at Woods Hole. And Salmon and Inoue are going to torture this egg, if you will, by applying high pressure to it. Ted Salmon developed this pressure cell where he could put hundreds of atmospheres very quickly and release it. And the pressure causes microtubules to depolymerize, and then when you release the spindle, they can polymerize again. And so if you watch the spindle here, it's low pressure, the pressure is applied, you can see the spindle dissolve, it dissolves within a minute or so, the pressure is released, it comes back. And so this ability to recover very quickly, I think, is very characteristic of a living system. I think there's going to be another cycle of pressure in a second. The pressure is going to go, or maybe the movie's looping. Anyway, there it is, dissolving and coming back again. I just absolutely love watching this. This is very similar to the egg extract spindles I'm going to talk about in the next talk, just in shape and form. It's a meiosis spindle in the egg. Here's another example. Whoops. This is two meiotic spindles brought in contact with each other in an egg extract. And again, the details don't matter here. I hope you can see the two spindles right at the beginning of the movie. It's a little more difficult here. They're first separate, and then they kind of jackknife together and then fuse. And I think this fusion of two structures into one, again, a bacteriophage certainly couldn't do that, is very characteristic of one of these self-organized assemblies. So I'll talk in more detail about some of the molecules, not too many molecules, in these spindles. Here I want to, I'll end the introduction by talking in rather general terms about the sorts of properties that make these spindles so lifelike, that allow them to, for example, recover from damage or fuse together in that last experiment. One important aspect is spindles are continuously exchanging subunits. In a way, first prove this, and then I'll show in my next talk some more recent experiments. But the tubulin subunits are continuously exchanging with the cytoplasmic pool, and one way of thinking about that is that the spindles are continuously rebuilding themselves, although they're at a steady state in size and shape that can last for many hours in the case of a meiotic spindle in an unfertilized egg. Every subunit in that spindle, except actually for the DNA itself, is cycling in and out rather rapidly. So I like to think of them as rebuilding themselves all the time. So it's not like the bacteriophage that builds itself and then it's finished. In the case of the spindle, it builds itself and it keeps rebuilding and rebuilding. And I hope you can understand that sort of makes it easier in some way for it to change shape or recover from damage. There are also a lot of mechanical forces in the spindle generated by motor proteins, and they're pushing and pulling on different parts of the spindle, and those forces have to be balanced at steady state so that things don't move. And again, the way I like to think about it is it isn't these forces act and then they're done. They're continuously acting, they're continuously kind of rebalancing themselves to get everything right on a timescale of seconds. And again, that helps it be lifelike. So continuous turnover of subunits and rebalancing of forces generate a steady state. That means constant over time, but still dynamic in both mass and shape. These processes require substantial energy dissipation. I told you about how microtubule polymerization uses GTP, but the motors in the spindle, I'll talk about later, burn a lot of ATP as well. So it costs biological energy to do this continuous rebuilding and rebalancing. But the net effect is an assembly that continuously renews itself and that can thus adapt, for example, to a changing shape of the cell, repair damage, and other lifelike properties. And let me end the introduction here. So here's this bacteriophage, a really exquisite biological assembly, so complicated, so amazing, does its job amazingly well, through a self-assembly pathway leading to an invariant size and shape. Here in the spindle, we're going to use this word self-organization to indicate assembly, but now to a steady state that continuously is dissipating biological energy to give a much more kind of lifelike assembly that can change and adapt and repair damage. Both beautiful biology. I'd say the spindle uses all the self-assembly principles of the bacteriophage, but has added to it this energy dissipation, this continuous change and rebuilding. And just to end here, although I've only talked about the microtubule cytoskeleton, I've really only talked about mitotic spindles, I think this principle of self-assemblies that continuously reorganize themselves, rebuild themselves, burning energy to do so is also true for the actin cytoskeleton, for the membrane systems of the cell, for the plasma membrane. Things like this are going on in the nucleus. Indeed, I think you could view the whole cell as a sort of self-organizing system along the same principles. So I think this way of looking at biology, which is, I'm sort of trying to share with you the way I look at biology through studying the spindle and microtubules, is a fairly general lens. And we'll leave you with that, and in the next segment we'll talk in more detail about how the mitotic spindle and the microtubule cytoskeleton at a more molecular way achieve some of these properties. So, thank you.